Good morning, everybody, esteemed colleagues, students. It is my pleasure today to introduce Professor Nicola Del Ferro. He's from the University of Padova, Italy. Professor Del Ferro is an associate professor in his university, and he focuses on soil and water management for agricultural systems, with a big emphasis in sustainability, in sustainable agriculture. His work is on soil properties that govern soil transport, nutrient cycling, carbon dynamics, and the implementation of soil conservation practices. In his talk today, he will speak about the fate of plant protection products like phytochemicals in agriculture, showing how specific properties that are very hard to measure, both physical and chemical, influence soil water vulnerability. Nicola is a very good friend. He's a colleague from previous projects. We have worked together very well, and I'm very, very happy to see him here. Nicola, the floor is yours, and thank you for joining.
Thank you, Yannis, for the introduction, and thanks to all for being here to this webinar session. Hope to keep my presentation simple enough for students and also interesting for faculty members. I will share my screen so that I can start my presentation. I hope you can see my presentation in full screen mode now. If it is okay, I can start. Then please, it can be, I hope, a sort of also discussion. There might be some questions. Then please feel free to interrupt me so that maybe we can also discuss about something of what I am trying to present. Okay. Then, actually, as you can see from my screen, I will discuss a bit of sustainable practices and monitoring and our results, our experimental results from a research that was conducted in the last few years here at the University of Padova, and also in our region that you will see, that is the Veneto region, northeastern Italy. As introduced, I am associate professor. I am also coordinating a master degree course, an international master degree course in sustainable agriculture, embedded in the Department of Agronomy, Food, Natural Resources, Animal and Environment. I am actually a soil scientist and agronomist. Then the question, let me remove, yes. As you can see from this picture here, taken from some NASA satellites during the night, we are here on the northeastern part of Italy. You can see that it is completely full of lights. That means that there is a high density. It is a very densely populated area with a lot of anthropogenic pressures over natural resources. And then it is also a very intensive agricultural area and also a very industrialized area. As you can see here from my slide, more than 350 people per kilometer square lives in the Po Valley. That is one of the most densely populated area of Europe. Then there is a lot of conflicts for soil quality, water, for natural resources with impacts on soil quality, water quality, and greenhouse gas emissions. And that is what we are trying to do also in collaboration with our Veneto government that manage the land and with also the other public administration. So then we provide support to the different bodies that manage the territory. That's why our focus is also on the water quality, and what I will present you today is something related to our results on the water, on the impacts of agriculture on the water quality. As you can see here from this picture, this map, you will see also in other slides, is our Veneto. It is our Veneto region. from northeastern part, not far from the Venice Lagoon and from Venice. And as you can see from this map with a lot of dots, blue and red dots that represent also this pie chart here, you can see that the chemical status in this case is of surface water is not that good, and it is not reaching the good quality that is required by the legislation by 34% of the sampling points. These data are not provided by us, but by the Environmental Protection Agency of the Veneto region that every year publish different reports. And this is taken from this website. Then this is something that is publicly available. Our approach, I mean, for sure, the water quality is not only due to pesticides, it's also due to industry, it's also due to nutrients and so on. What I will focus here today is about the impacts that can be due to agriculture in terms of phytochemicals in surface and the ground water. Our approach is multi-scale because we have seen over the years that the impacts at the territorial scale is also due to micro-scale characteristics. So that I will show you something related to also micro-scale analysis that can affect the macro-scale results. Then the main factors that we identified as affecting the quality, especially in our area that can be vulnerable to contamination, is that there is a highly intensive crop production system. We have also very shallow groundwater tables that can be just below 50 centimeters, 1 meter, 1 meter and a half. And the importance of water reclamation companies is well known in this area. Another important aspect is that we have loose soils with poor structure, with low organic matter content that sometimes cannot be very, cannot be so good as buffering systems as protection for groundwater contamination. Then our approach, you will see, is going from the territorial level to the lysimeter scale results and also to the micro scale analysis. Then our first open questions that were then published in some papers here in a Journal of Hydrology, and I will show you something about our results that were published here, where in this research we were trying to identify if the shallow groundwater is a driver of pesticide contamination, then if the pedoclimatic conditions can affect the movement and the impact on the groundwater, and if different agricultural systems affect the pesticide movement, and then what is the contribution of the different agricultural management practices. And what we found is then reported in the few slides that I will show you later, with some concluding remarks that I hope can summarize our results. We work at the lysimeter scale in this experiment. A lysimeter is a system like you can see here on the left, that is a closed system representing is a sort of mesoscale that is a bit in the middle, the laboratory analysis and the field scale analysis, with the advantages that we can have very high controlled conditions. From the picture on the right, you can see also some rooftop that can move and that we can, for instance, avoid rainfall in our lysimeter system that is composed, as you can see on the D image, by 20 lysimeters. They are closed on the bottom, and in this lysimeter system we are monitoring soil moisture, matrix potential, and we are also able to collect the pore water at different depths and also at the groundwater. The system is fully automatized so that we are able to regulate everything and then collect the water that is moving through the soil profile automatically. And at the same time, we are able to monitor at the same time then the soil moisture, matrix potential, soil temperature, and so on. Then in this way, we were able to characterize the soil profile. And then we were able to characterize also with some tracer test the movement, in this case of bromide, just to understand what was the movement and if the different shallow groundwater conditions that we managed differently in this lysimeter system could affect the solute transport. And our approach is a combination of experimental results with modeling results. In this case, you can see some, I won't go to the details of the results. It's just to show you that in terms of water content and in terms of pressure head, then hydrological and the physical conditions of the soil during our experiment, we were able to monitor these conditions in terms of dynamics of the soil, and we were also able to model very well the soil conditions. The approach was the inverse method that for some is probably very clear what it is, for some others not. The inverse method is a way, in this case we use Hydrus 1D, hydrological model. In this way, with the inverse method, we were able to somehow reconstruct or characterize the soil properties based on the results obtained and based on the boundary conditions that were very well known along the soil profile. After that, after the characterization of the soil hydrology, we were able also to characterize the solute movement. Here on the graphs on the left, you will see some examples of what was the movement of the bromide at different depths, then at 15, 30, and 60 centimeter depths during around 200 days of experimentation. And then we were able also to collect the groundwater we were able to discharge or recharge according to capillary rise. Anyway, we were able to characterize also in terms of mass the movement of the solute with the water. And we use different modeling approaches just to describe the solute movement with two approaches. The simplest one that is called advection dispersion equation, where the solute is moving with water, then it is the simple way to characterize the solute movement just with a dispersion coefficient and diffusion coefficient. I will not go into much to the details, but with this simplest model, we were not able to fully characterize the movement of the bromide. Then we try to skip this approach and to go through a more complicated one that is called the mobile immobile model, that is used to characterize non-equilibrium flows, meaning that some preferential pathways could occur. And this is what we found. We found that what you can see here on the left, then the experimental results were well described when the mobile immobile model was used also in this case with Hydrus 1D. Then our results was that yes, some preferential flow can occur even if there were not some visible cracks on the soil, but probably due to some macropores. And then what we did is try to combine this modeling evidence again with some other experimental evidence. so that we went through the legimeters, we collect some small samples, and then we scan with X-ray microtomography. And you can see here the picture on the right, what it means. Then we have an X-ray tube that is then, I mean, it is then analyzing the sample, and then the projections are collected in 3D in order to have a 3D reconstructions in an undisturbed way on how is the soil structure. And then on the left side, you will see different images. From the top are the slices, then like the different pages of a book in the 3D dimension. And the different letters, the different labels, FD, WT, and WT 120 and 60, means the different water table levels. That is free drainage in the first case, a water table, a shallow water table put at 120 centimeters depth, and 60 means at 60 centimeters depth. What is evident, even from the first images on the top, is that when we have a shallow water table, we found that it was more frequent some macro porosity. Up to, you can see here from the second image that is colored, up to more than 2 millimeters, 2.8 millimeters of macropores that were frequent with the shallow water table and were not frequent in the free drainage. And then we combine these results, these experimental results, with the macropore result, with the modeling results. And our concluding remarks from this experiment is that the shallow water table actually affects the soil structure. And in turn, it governs some preferential flow pathways that can make the groundwater vulnerable to contamination. And then the fast movement of the solutes through the soil profile increased with an increase in frequency of macropores. Then, since in our region, one of the ways to provide water in the summer period that can be very, very hot, very dry, is also to use the management of the groundwater. And then to provide water through capillary rise, this can be very good, but it can also create some vulnerability of the groundwater for pesticide or other solutes contamination. Then this is our first, results that these are the first results that I wanted to share with you. And then we go through another experiment that was still conducted at the lysimeter scale, but with a different approach, because we discuss a lot about how to shift intensive agriculture to more sustainable practices. And one of the ways is also the conversion to conservation agriculture in croplands. Conservation agriculture is also supported by the CAP, by the Common Agriculture Policy, and also supported then by the Veneto region. And for us, conservation agriculture means no tillage, the application to then the crop rotation and the use of cover crops throughout the year. However, especially in the low-lying plain, in this silty loam and the loam soils, it is rarely adopted due to the predominance of these soils that are prone to compaction, particularly under no-tillage conditions. And then the idea is, can we find a compromise? Can we support conservation agriculture and avoid some compaction? Then one of the approaches that we were trying to adopt is to use some peculiar cover crops like tillage radish, that is also called forage radish, that could be used due to its very big taproot apparatus that could alleviate soil compaction and then improve soil aeration and regulate water and nutrient cycling. However, under no tillage, there might be also a sort of biological drilling that could be also increase water and solute vertical movement and then create preferential flows, maybe after the vitalization or maybe also during the crop growth, as you can see from this picture on the right. Then this was the topic of our research. And then try to estimate water and solute transport dynamics during the growing season as affected by the taproot induced structural changes, and then understand the vulnerability of the groundwater to contamination in this potential agroecosystem of northeastern Italy. This was also published recently in a paper in Soil Science Society of America Journal. And then I will present some of the results, and I'm open to discussion with you if you have something to ask. Again, we try to combine both experimental and modeling results. In this case, for instance, we work also at the small scale in pots where we grow forage radish. You can see here from the picture on the left. According to collaboration with some colleagues from France, we were able to collect some projections, as you can see here on the picture in the middle, that you can also see some cracks due to the root apparatus that was growing during the season. And from the right, you will see in white the root growth that is growing. And then around you will see in yellow the porosity, the macro porosity that was affected, and you will see that was actually affected by the root growth, while the dark part is the soil matrix with only micro pores. And indeed, what we found in terms of results is that, as you can see from these pictures, for instance, on the left side end, you will see the pore distribution after 60 days of growing with respect to the pore distribution at the T0 without any growth of the root apparatus, that is the green part. You can see that the porosity, the macro porosity especially, it is increasing, especially on the top, where we had more roots, especially the taproot apparatus. This can be combined with the results, as you can see on the right, about the 2D root diameter that is increasing, especially in the first centimeters after 60 days. And this was also validated by this relationship in between the 2D root area per slice, or the 2D root diameter, and the 2D pore diameter. Then the increase in the roots was also associated with an increase of the macropore in the soil profile. And then we went through the lysimeter experiment again, as I showed you before. You will see also later some pictures about the experiment. And then our question was if differences occur in water and solute transport dynamics affected by the taproot induced structural changes. And again, we combine a tracer test with, again, modeling to evaluate the solute dynamics. And what we observe here are some pictures, for instance, related to the bare soil on the top, again, with the free drainage or the water table or a shallow water table. And on the bottom, when we use the taproot, both in the free drainage and with the shallow water table at 60 centimeters depth. You can see that even in terms of dynamics, there is a large peak, especially with the forage radish, with respect to the bare soil. Although we did not find big differences in terms of hydraulic properties that were slightly affected by the tillage radish compared to bare soil. The major changes concerned an increase in the interaggregate porosity, then the macro porosity. Then due to the fissures and channels that we also demonstrated to increase with the tap roots, that also increase along the leaving roots that induced some non-equilibrium solute transport. Then some preferential flow still occur and was found with the taproot. Then the question is, is it possible to use this cover crop, or is it increasing the vulnerability of the groundwater? While actually, as you can see from the pictures on the right, during the growing season, then the taproot was growing very well, and the taproots was, deepening down to 40 centimeters depth. And we observe also a lot of biomass production at the different water table levels and also with the free drainage. That was, associated also with a very high uptake of the solute. Then it means that, yes, might be some preferential pathway, but actually we did not observe that much the release of bromide because the vegetation was able to catch the bromide. Then it acted as a catch crop, as also demonstrated here by these results provided by experimental data and also supported by modeling results. Then, as you can see, the uptake of the bromide was up to 48% with a shallow water table of 120 centimeters depth. Then it means that a lot of the solute was actually uptaken. Then, what about the vulnerability of the agroecosystems under different water table management? Is it vulnerable or not? Then, what we can say, and this is our concluding remarks about this second experiment, is that the leaching was actually offset by the taproot system that served as a solute scavenger, despite the macropores increased. However, the tillage radish in conservation agriculture is usually devitalized. Then there is a devitalization also with some herbicides. Then there might be the release of previously absorbed solutes and the creation of also wider biochannels, especially in no-tillage system, due to, the degradation of the biomass of the cover crops. Then there might be this, and we did not investigate that much. We are waiting for some funds, for some other investigations. But this is what we think that are our main outcomes from also this second experiment. We not only worked with solutes, with tracers, but we are also working with pesticides or herbicides, plant protection products in general. For instance, some works we are conducting with glyphosate in different areas, both in croplands and also in the hilly areas where you may know that it is very intensely cultivated, for instance, for Prosecco wine production that is very well known worldwide nowadays. And then we investigated again the effects of conservation agriculture combined with the shallow groundwater conditions on the glyphosate movement. And also we investigated the movement of glyphosate in the north, in the hilly areas where it is used for devitalizing on the row the grass in vineyards. And actually glyphosate is an herbicide that is very well absorbed to the soil particles. But what we found is that from a laboratory point of view, yes, the glyphosate is well absorbed to the soil. But in practice, what we observed is that at different depths, as you can see, for instance, with the diamonds in white here, even after a few days after application, we can find it at 60 centimeters depth. Then again, we found some fast movement of the glyphosate. This is conducted on the top left again on the lysimeter experiment. But what you can observe also on the right side end is that we found the fast movement of glyphosate also in field conditions on grasslands and vineyards monitoring studies. Then again, you can see here from some experimental results that we found also, especially glyphosate and also AMPA sometimes also at the bottom. And as you can see here from the right, we also found glyphosate again in groundwater. Then some susceptibility of the groundwater to contamination can occur. What about the fate? Then we see that can move easily. Not always, but especially after some intense rainfall events. Then some good practices are also provided, and some guidance can be provided to farmers how to use that, not only in some specific soils or not in some other specific soils, but also based on climatic conditions. And what we observe is also anyway that the degradation is quite fast. And what we found is that our results are in line with what can be found also in the literature. For instance, in terms of dissipation with half-life, we found that it was in between 30 and 40 days in terms of dissipation of 50% of the glyphosate in the soil. That, of course, dissipation is not only degradation, because dissipation means that can also be leached out or can also move to the surface water after some runoff and soil water erosion. Again, some results from our experiments at the lysimeter scale under conservation agriculture, because glyphosate is used for the vitalization of the cover crops, especially in conservation agriculture, where we cannot till the soil, and then it is also used for weed suppression. Again, the approach was the same, of combination of experimental and modeling results. What we observe is that actually, just to summarize in one slide, is that it was not possible to estimate the movement of glyphosate with the mobile model, as we have observed, for instance, for the tracer test. But the approach required was that of the dual permeability, meaning that our soil is composed of two different systems together. Okay, one where the water flux is mainly moving, and the other one, a little part, where the solutes can go through and then reach the bottom layer or the groundwater very fast. And indeed, it is only with this approach, with the dual permeability model, that we were able to simulate what was observed also experimentally. Then still, our main conclusion is that glyphosate movement was very fast, sometimes can be very fast, and can reach the deepest soil layers despite its high affinity for the soil particles. And the conversion to conservation agriculture in our short-term experiment did not affect actually the results, but we were in a transition period. Then can be that in the long term we have different results, maybe also a mitigation effect. And if we wanted to model and predict the movement, we must apply complex models, complex algorithms such as the dual permeability to describe the experimental results and then predict the experimental observations both in time and in space. We are then now using, for instance, some amendments that can be used, for instance, in vineyards such as biochar, to increase the absorption rate, not only of glyphosate, but also of other fungicides such as fluopicolide, in order to evaluate if we can somehow mitigate the movement of plant protection products in agriculture. Last, this is just last. I have just I have some last slides, 3, 4. Based on this, we are trying to upscale our results to the entire region. Then the approach is to combine some biogeochemical models. In this case, we are trying to use the EPIC model. And then the combination of this model at the territorial level must be conducted with data from the management, from the soil, from the climate, from the pedology, and so on, in order to create homogeneous areas where to evaluate the movement of pesticides. Why are we using biogeochemical models now and not hydrous, for instance? Because our idea is that according to an holistic approach, we must integrate the movement and the predictions of plant protection products with the main biogeochemical cycles, such as carbon, nitrogen, and phosphorus. Of course, the model, I will not go through what is EPIC. It is, here reported, and then I don't want to go too much to some details. What I wanted to emphasize is that we implemented a subroutine that we call pesticides that somehow was able to include in a more complex way, for instance, the preferential flows that was not embedded at the beginning in the EPIC model. We also implemented some other algorithms in order to improve the wash-off effect, for instance, the non-linear absorption of some plant protection products such as glyphosate, and also the half-life time based not only on some numbers that we can find in the literature, but also based on the soil conditions such as temperature and moisture. Our first results, they are not yet published. We are now completing a paper with a PhD student working with me. Then the idea is to publish soon these results that are very interesting because we were able to simulate what we found according to the experiments, so that we were able to simulate again the fast movement of plant protection products, also of glyphosate. And the work in progress is then to extend these results to the territorial level and to associate this with experimental results provided by the Veneto region, also at the catchment scale and not only in specific experiments. Then my general conclusions are that we must have targeted application both in time and space to reduce the risk of loss to surface and also to groundwater. And then that plant protection products such as glyphosate, theoretically of low risk of leaching, can actually impact the groundwater. Then the preferential flows, the preferential pathways can occur and should be taken into account. and that the modeling tools are necessary not only for the prediction, but also for the monitoring, the verification, and the understanding of the best practices to be adopted. I am done. I have only to tell you that these are the topics that we are also trying to transfer to our students, to our international students in sustainable agriculture. That is an international course where that is completed, that is completely in English. We have different curricula. We were discussing also with Yanis about some possibilities of collaborations together. It is embedded well within sustainable agriculture. We had also some dual degree programs with the United States and also with Erasmus Mundus. And I mean, we are open to collaborations in different topics, such as what I have discussed with you. And that can be maybe, and there might be also some other room of collaboration together on this topic or in other topics. Then I'm done, because otherwise I am, I mean, going to, I mean, I am talking too much. Then if there are questions or I am open to discussion, then thank you.
Thank you very much, Nicola, for this very nice presentation and really exciting new approaches regarding sustainable agriculture. Now we have some time for the Q&A session, and I already see your hand raised by Professor Petridis. Yes, please.
Thank you, George. Thank you, Nicola, for the presentation. I have some questions, but I would like to let you inform that it's from the curiosity, because I'm not coming from your topic. The first question that I have is like your sensing systems. Do they provide any selectivity? Because you mentioned about bromide, if I got well. I mean, within the soil, probably there are other kind of elements that you would like to stop or to detect. So what kind of selectivity, and if you provide any selectivity, how can you manage this?
Then the system is able, the system can, you mean selectivity that, well, we are collecting the pore water, okay? Then we could also have analyzed, for instance, nitrogen movement, okay? But the topic was about only the bromide, because we were sure that there was not bromide in the soil solution, okay? Then what was moving was just from what we were distributing, what we were applying from the top, okay? What can be tricky sometimes is not related to the bromide, okay, but to some molecules, organic molecules, such as plant protection products that can be somehow not, I mean, that can be blocked, okay, by the system that we adopted, that are some suction cups, ceramic cups, suction cups that are in the soil, that we were able to test specific suction cups that are not in ceramic, but are in plastic fiber, that can, I mean, be non-selective for these specific pesticides. Because otherwise we were somehow saying that, yes, there is not the plant protection products within the soil profile, but actually there was.
So if I got, well from your answer, like how do you stop, the detection of the other kind of elements?
We are not stopping. We collect, but we only analyze what is of our interest.
What method do you use in order to analyze, only the bromide?
Well, it's a chemical analysis of the bromide in soil solution.
All right, chemical. So you're using chemical methods.
Of course, of course.
I mean, you are not using
No, we collect, yes, no, we collect the sample, yes.
All right. Okay. So my second question has to do, since you are moving, I like it very much, your approach that you are moving from the micro to macro. This means that probably you have a lot of space to develop prediction models. So like if in the nano scale that we say in physics or in your scale at the beginning, so the concentration of some elements that you can detect in micro can also lead to prediction models instead of, I mean, if I detect this in this soil, then, in a micro, I have this problem, or you can use this nano or micro detection for prediction model that something starts to be involved, and then I need to act before, a whole damage. So
I mean, they are different models, okay, because there are also poor models, okay, but actually they are, according to my experience on that, I had some collaborations with some guys from Mexico and New Zealand, and then we used it was called smooth particle hydrodynamics, a specific model to simulate exactly the pore movement, okay, but we were able just to simulate small samples, small soil samples, okay? Of course, they can be used to evaluate what is the role of the macro or the micropore, okay? Then they are fundamental, I would say, for the understanding, okay, of the processes. Then could be interesting according to your suggestion and question. I take it as a suggestion, okay? Also to evaluate, for instance, or to, actually evaluate whether the solute is actually moving on that macropores and not in the micropores, okay? Then actually we use a different scale, okay, of modeling, but could be of interest, of course, also that kind of approach, and then to combine together.
I would say, following your approach that you are doing simulations and also you are doing simulation to compare your experimental results, I would say also the AI, I was thinking, will play an important role for your prediction. I repeat, prediction, all right?
Yeah.
Like, if you detect, let's say, if we take the example from cancer, that you
of course.
small thing, that the problem will be there.
Of course.
The cancer science and research that they detect small things in order to predict what it will happen later. And my last question, if I will allow, George, you mentioned about nonlinear absorption. So coming from nonlinear optics in lasers, in our case, in order to induce nonlinear mechanisms, we need, high intensity. So just from curiosity, the nonlinear absorption mechanisms, can you please describe the mechanism? What do you mean by nonlinear and how?
Nonlinear is not absorption, chemical absorption, but is about, it is the equilibrium of the molecule, okay, in between its affinity for the soil or the water phase. Okay, then some concentration, usually there is a sort of saturation, okay? Then you can have some solubility of the molecule in the soil, in the water, okay? But it has some affinity according to chemical physical characteristics for the soil particles, such as clay or silt, not usually sand, okay? Then there is some more or less affinity, but there is some saturation threshold, okay? If the concentration is very high, there is no more affinity for the soil particle, and all the solute will come in the water phase, okay? Then glyphosate has this specific, it's typical of organic molecules, okay, that have this non-linear equilibrium in between the water and the solid phase, okay? So that at low concentration might be, for instance, 50% in soil, 50% in water, let's assume, okay? But if the concentration is high, it is not possible to have 50% in soil or 50% in water because the soil is fully saturated, okay? The exchange sites. So that maybe you have 40% in soil and 60% in water. I am really simplified a lot, okay? But just for your understanding, okay? If the concentration is even more, okay, you have lower content in the soil, maybe 20%, and then 80% in water. So that you see that this equilibrium is non-linear.
Okay, thank you. I will need, to study a bit,
The approach, you can check for Freundlich isotherm. That is one
Yes.
of the models used to evaluate the equilibrium between solid and water phase.
Yes, and your work with Yanis for these joint master courses and joint courses, please go ahead, because I think that this is one of the most sustainable internationalization. Since we speak
Thank you.
about sustainable agriculture,
Yeah,
let's speak
of
about
course,
sustainable
of course.
internationalization. Thank you, Nicola.
Thank you,
Very much. I
Kostas, for
appreciate it.
the questions, and Nicola for the nice discussion and the insightful feedbacks. And then I will move to, as I see in my screen, Bogdan. Yes, please.
Yeah, thank you. Thank you for a very impressive talk. I thought it was like 5 minutes. I haven't noticed how we spent like 45.
Thank you.
There were a couple of very interesting parts in it. I also used my time yesterday and I found some other ones that you also published. For me specifically, I'm also not coming from agricultural approach. I'm computer scientist. I developed some applications in a cooperation with agronomists. And for me, the most curious part is do you share or do you publish any of the data you have? I found some of those topics very interesting, especially for this time series data or for this geospatial data. I found some other ones of yours with hyperspectral imagery, and that would be very interesting to dig in to try to reproduce some of your work. If you could lead me, I can write you after the talk. And
Yeah, let's see. I mean, what we publish, what you find is what we publish.
yep.
And then if you are dealing with some possibility of collaboration, of course, yes. We are also working, as you mentioned, with hyperspectral data. It's completely another topic.
Yep.
With also AI, as mentioned by Konstantinos, because it is required also this approach when we have a huge amounts of data. And we are working with, especially now, a PhD student working with me is working on fertilizations, for instance, and the prediction of the requests of the vegetation based on the stress, based on the response of hyperspectral data. Then it would be great to have some form of collaboration, because we have started this project just last year.
That would be perfect. Yeah, we also have some remote sensing applications for soil content analysis for other properties, but what I found really impressive, this dynamic approach is, it's not that easy with remote sensing applications.
Yeah, indeed. In that case, yes, we use data from storm events. Yes, that was a very big topic.
Yeah,
yes.
so thank you very much. That was a good time.
Thank you. Thank you. Thank you.
Thank you both, Dan and Nicola, for the nice discussion. And then I will move to Ioannis Luludakis. Yes, please.
Hello. Out of curiosity, you may have mentioned this, but in your experience, is it more about the actual active ingredients, or is it about the helping agents? About the movement, I mean.
Can you repeat? Sorry, if it is.
You were detecting the active ingredients, right?
Yes.
So bromide, let's say, for example. But the movement, is it more about the active ingredients, or you found it in isolation, or is it part of the solution, let's say? So it's more about the helping agents.
I mean, it is both, because it depends on, of course, about the physical-chemical properties of the molecule. More absorption, less absorption, but not only. Because as you have seen, for instance, about some plant protection products, because, for instance, bromide is fully in water. It is not absorbed in the soil particles. Then it is easy to understand, yes, it is moving with water. But not for some plant protection products that are developed to be kept in the soil, okay, and not to leach out. Then in that case, the problem is that, or sometimes it might be some issues and impacts related to some specific soil properties, such as macropores that can, create, or biopores that can increase the vulnerability of the groundwater to contamination. Then it is both. It is both. I don't know if
but well, yeah, but you didn't detect depending on the helping agent. Right?
Sorry?
You did not. Can anyone help me?
Yeah, not very well.
You cannot hear me?
Yes, but it is a bit disturbed.
Sorry.
Nicola, I think
Sorry.
Yes.
What Yannis is trying to say is that if you try to detect also the substance where the bromide was dissolved.
It was dissolved. I mean, it was dissolved in water.
So it was only water. It wasn't.
Of course. Yes. No. Sorry, Ioannis. I didn't catch your question. Yes, it is only dissolved in water. Yes.
Thank you, Ioannis, for the question and Nicola for the feedback. And then Yannis Daliakopoulos.
Okay, let me say, first of all, that I'm so excited about this presentation, also because I can hear things that are very close to our research. Also because I see that you're using micro-CT scanning, and of course I knew this already, but we in HMU in September we are going to have a very nice micro-CT tomographer, and we are very excited to see the results and what's going on. We're also working with scimeters, but of course not at the scale that you are doing right now. But it's in the plan to develop an installation that's, trying to approach what you're doing. I have one question on the use of hydrous and how you modeled what equations you used to model the movement. So was it Wagenknecht? Was it something else?
Yes.
First of all. And if what you're saying is with two types or two velocities, let's say, of movement.
Dual permeability, yes.
Yeah, if it was modeled by modal equation or in some other way.
Yeah. Then what is interesting, thanks for the question, Yannis. Then what we observed and what is interesting is that the movement of the solutes has a different dynamic from the water. Then, for instance, when we model the bromide, we used with hydrous the mobile-immobile equation. That means that there is some exchange between some solute that is, somehow kept on the soil and some others that is moving with water, okay? So that you have a non-equilibrium flow, okay? And what is interesting is that this approach was only required for the solute and not for the water, okay? Just for the hydrological part, we use the simplest Wagenknecht-Mualem approach, okay? Regarding glyphosate, according to the absorption coefficient that we measured, okay? Let's consider that can be 40-50, the repartition between soil and water. That means that all the solute in theory is in the soil, and this is what we measured, okay? Then according to that, also the mobile-immobile was not sufficient in order to simulate the movement of the solutes, okay? Because, it is kept all in the soil matrix, okay? So that we adopted the dual permeability. That means that you have a sort of, you are, reproducing two times the hydraulic properties and the solute properties. One for the macroporosity and one for the matrix domain, okay? It is actually more complex, and you have more parameters to calibrate, okay? That is very good for laboratory or lysimeter approaches. If you want to upscale, for instance, at the territorial level, it is very, very difficult because you don't have data. That's why we use some simplest approaches, okay, such as the biogeochemical model, with simple algorithms to estimate some crack flow. I don't know if I answered to your question. And actually, what we used is usually the inversion, because we have all that amount of data. From those, we can somehow build our soil properties, able to predict the movement of the solids and also of the water.
Okay, so you didn't consider getting a sample of the soil and measuring the properties, let's say, with high prop.
We also did. The problem is the heterogeneity. Always,
Yeah, okay.
because for
But-
instance, you have seen that we have collected the samples. I did not show you, but we have also the soil hydraulic properties of those small samples. But we have 4 centimeters. Okay, then to use that one for the lysimeter scale can be helpful, but especially for the calibration, but not for the exact condition. At least this is what we observe because of the heterogeneity.
Yeah, of course. Okay, so it's a different scale again. You're coming from
I would say
okay.
yes. This is tricky to shift from one scale to another.
Okay, excellent. Thank you.
You're welcome.
Thank you, Yannis, for the questions, and Nicola for the nice discussion and feedback. And I see again Ioannis Luludakis. He has some additional comment or question.
Oh, no, sorry. I just forgotten to lower the hand. Sorry.
Konstantinos Petridis.
Yeah, I have an additional question to Nicola. Listening now and thinking of his presentation that you are measuring, you mentioned kinetic dynamics. So you have bromide within water, all right? So the kinetic dynamics are very much affected by the mass of the molecule that you detect. What about aggregation of the bromide particles? I mean, how this aggregation, or if this is the case, affect your kinetic dynamics, and how you can distinguish that you don't measure a kinetic aggregation due to the kinetic dynamics, I'm sorry, due to the particle aggregation, so you like the bromide particles to collide, to stack each other. How can you distinguish this?
I mean, honestly, we didn't get to this point. Okay.
All right, so you don't have an aggregation of the particles.
No, because I mean,
so
it is widely used as a tracer. Okay, it's
Alright,
like it's like chloride. Okay, it is fully in water. Of course, chloride can somehow aggregate with sodium. Okay, but bromide is usually used as a tracer that can move just with the water phase. Then it is not affected by any aggregation. Okay. Then we can say that it's like nitrate moving in water in agricultural systems. Then it is just moving with the water.
All right. Thank you, Nicola.
You're welcome.
Thank you, Kostas and Nicola, for the nice discussion. And yeah, I think more or less we reached to the point where we can finalize and conclude this presentation. We are very glad and very happy to have you today with us, Nicola. This was a really nice contribution, proven by the engagement and interest of the participants during the Q&A session. And before we close this, I would like to announce next week's presentation. It will be given from Professor Konstantinos Adreadis from the University of Massachusetts in the United States of America, and he will discuss his research about big data in hydrology, opportunities and pitfalls. Until next week, take care and see you soon. Bye.
So let's mention something that the next week's presentation is very much linked with what did we propose or what was discussed with Nicola. How do we use big data in this topic? I think that is very much linked. So Nicola, you are more than welcome to join.
Thank you. And thank you for the invitation. And thank you for the invitation and keep in touch.
No.
Bye-bye. Have a nice day.
Thank you very much.
Take
Bye-bye.
care.
Bye-bye.
Bye-bye.
Bye-bye.